Okay. Start. So, welcome everyone to today's Phytochem Talks. Before I introduce the speaker today, I have some announcements to make. First of all, this is a reminder that the Phytochemical Society of North America meeting is in from June 15th through 19th at the University of Wisconsin-Madison. The registration is open and early bird rates available now. So please do register at just search for PSNA 2026 and you'll get to the website. There's also the International Society for Chemical Ecology meeting at Cornell being organized this year. We purposely coordinated with PSNA to keep the dates non-overlapping. So the date here is right after PSNA. Cornell and Ithaca are beautiful in the summer. So it's a great opportunity to visit upstate New York and look at all the beauty nature has to offer in this part of the world. There's also the Metabolomics Association of North America meeting. The registration is coming soon, but this will be at UC Davis from September 8th through 11th. This is organized every year. It's the 8th annual conference, and yeah, there are very interesting talks being scheduled, not just in phytochemistry, but also regarding metabolomics in general in human health, bacterial metabolism, fungal metabolism, etc. So please do keep this on your radar too if you're interested. Okay, in the coming days, while you are waiting for those meetings, we also have a continuous schedule of Phytochem talks. So in the coming days, we have Design Ding speaking on medicinal chemistry, Adriana Lopez in early April, actually talking about exploratory studies of in vitro plant pathways, and Lorenzo Caputi will talk on single cell metabolome and RNA-seq analysis. So today's speaker, it's my great pleasure to introduce Dr. Sushma Naithani. Sushma did her PhD in botany from the National Botanical Research Institute in India. And then coming to the US, she was a postdoc and research associate at Iowa State University and then at Cornell University, where she also then lectured in plant breeding and genetics. She then got a position at Oregon State University and moved on as an assistant and associate professor, senior research. And now she's been teaching several courses in botany, plant breeding, and genetics at Cornell as well as at Ohio State. She's published several papers on many of the important resources we use as senior in Gramene. She is an active contributor to the Gene Ontology Knowledgebase, Gene Ontology Consortium, as well as done some work on Plant Reactome that we'll be hearing about today. In addition to all the research that she's been doing, she's a very active member of the overall plant science community. She's worked on biocurators as a biocurator and the steering committee member in several important databases. She's reviewed over 800 manuscripts for 20 journals, and it's likely that one of our manuscripts were reviewed by her too, as well as served as a panel member for several federal organizations. For several years, she's been organizing the Plant and Animal Genome Workshop on Systems Biology and Ontology every year in San Diego. So if you happen to go to the PAG meeting, you can meet her in person. Over the years, she's been awarded several awards, including the AgBioData Mission Leadership Award, Excellence in Biocuration Award, the START Superhero Certificate for Advising and Mentoring from Oregon State University, and the Excellent Scholar Award by the International Botanical Congress. So without further ado, I'd ask Sushma to share the screen. She's going to talk about the Plant Reactome knowledge base.
Okay. Are you able to see the correct share?
Yes.
Yeah.
That's correct.
So, hello everyone. Thank you, Gaurav, for the nice introduction. and inviting me to Phyto Talk. So this is a great opportunity to talk to you who are in the phytochemistry community. And I did not realize that we have these parallel communities and we have no outreach here. So this is really a good opportunity. So today I'm going to talk to you about Plant Reactome Knowledge Base and little bit about the other databases as well, as we are working in the ecosystem of the public genomic and genetic and breeding databases and how Plant Reactome fits there. So just as an overview, so we have like since past 25 years, a lot of genomic data, transcriptomic data, proteomic and metabolomic data. So overall, our community, as you know, has two major conceptual frameworks. So one framework is the genome browser, and other framework is the pathway databases. So of course, both of these frameworks allow us to integrate heterogeneous data that comes from various techniques and various studies and synthesize knowledge. So typically, genome browser allows us to map the gene models, look into the genetic variations, look into the transcriptomic patterns during uh, any organism's development or response to the environment, and so on. And it allows us to connect genes and proteins and transcripts, gene expression data, and pathway databases, on the other hand, allow us and in a way complement genome browsers in a way that they can connect the genetic data, that is most of the gene models that are encoding for proteins and RNAs and all kind of regulatory molecules, but also it can connect to the proteins and metabolites downstream. It can also see how these genes influence metabolite and biochemicals, and together what they meant for organisms' development, phenotypes, traits, and so on. So this is where we fit here. Plant Reactome is again a pathway database and it connects to genome browser database, mostly ensemble Gramene, plant genes and gene browsers. And then, it connects to all those kind of the uh, other type of the data as well. So anyway, when we do we develop genome browser as part of the Gramene, or we develop the pathway databases such as Plant Reactome, we are continuously doing gene annotation. We are also, especially with the pathway databases, looking for the gene interactions and how genes are associated with various biological processes, pathways, how they influence traits and phenotypes and molecular function in relation to plant development and response to environment. So overall, Plant Reactome is a reaction network database, and it explicitly describes biological pathways as a reactions that transport, transform, or reorganize physical entities. So we have many different type of the reactions, as you can see, that can be described or represented in Plant Reactome, and we have many different type of the entities that can serve as an input, and we can have various different type of reactions, including metabolic reactions, but also gene regulatory reactions, also transport and translocation reactions, and so on. And we use the Go associations. We have unique names for the pathways and the events and molecular entities. So we are interoperable and we work in the ecosystem of the public resources. Overall, Plant Reactome, a little bit about its history. So Plant Reactome basically began as a pathway portal of Gramene database. Before Plant Reactome, we have developed rice cyc, maize cyc, sorghum cyc using the BioCyc platform. And in 2012, we moved to Plant Reactome. And so Plant Reactome initially, obviously, we started with metabolic pathways, some of which we imported from the rice cyc. And then slowly we started adding other type of pathways, including hormone signaling. And then we slowly looked into how to represent plant processes related to plant development and how plant respond to biotic and abiotic stress response, or for example, stimuli such as gravity, and started curating those processes. So typically, this is a production overview of plant reactome. So we use reference pathways that we manually curate, and then we use gene orthology based projection of these reference pathways and have the projected pathways. For each release, then we combine reference and projected pathways, and plant reactome production is made and made available to public. One can access it for free of cost. One can also query manually one by one, or one can use API for downloading the bulk data. And this also allows users to not only browse the pathways, but one can upload their own data. It could be transcriptomic, metabolomic, proteomic data, and do the pathway enrichment analysis, or simply do species comparison, how different pathways and reactions diverge or are conserved across the various species in the plant reactome. So this is the website of Plant Reactome, so plantreactome.gramene.org. And currently what we have rice as a reference pathway, rice as a reference species for which we curate uh, evidence-based reference pathways. And we have gene orthology-based projections for another 138 species. This data is again open and it is free of cost, and it is implementing fair data practices. We have training videos and tutorials available. And here we have like two ways you can access. So one can search for entity or protein or metabolite and can land into a specific pathway. But other way, you can also browse, and this is what you land up here. And so typically this is the overview of the pathway representation in the Fireworks. So we use Neo4j platform to host this data. So bulk of our pathways are still metabolic pathways, but then we have pathways in other categories as well. On the left side, you can see the hierarchical listing of the pathways. By default, we show rice as a reference species. And typically, if you choose any of the pathways from this list or from the graph, one can land into a specific pathway. Here is an example of a signaling pathway, salicylic acid signaling pathway. And here one can see how salicylic acid biosynthesis pathway is connecting to the signaling pathway. So these pathways are basically divided into cytosol. The metabolic pathways and the signaling pathways are taking place in nucleus. On the bottom side, we have description, which can have pathway summary with citation and links to PubMed publications. And then we have all other categories, molecule listing and their mapping and so on, as I will show you. If you are interested in a species that is not rice, then you can browse from this list of available species and jump to projected pathways. So for example, here is Arabidopsis, and one can see Arabidopsis pathways. Other part of Plant Reactome is that how do we integrate omics data and provide additional information. So what we have collaboration with EBI Expression Atlas. And if you select any protein or gene product in the pathway, you can also bring the gene expression data that is coming from EBI Expression Atlas and can see how tissue specific expression of this gene occurs. So this is kind of a remotely fetched data that is coming from EBI. So that is one way of integrating the omic data and provide enrichment of the pathway information. One can also look into other type of data. So sometimes in the pathways, you will see certain gene products or the proteins that have these kind of the icons here. They have certain numbers, so it means that some of the gene-gene interaction data is also being projected and is available for that. So I'll go into that later. And then overall, this bar here that you can change the view on the bottom panel uh, tries to map and provide the links to complementary information that is available in other databases. So Gramene, for example, for the gene, UniProt for the protein, and CABI for molecules, and PDB for the protein structures, and so on. So again about this interactor mapping. So these interactors are not curated. So they are being projected from bar and some of the INACT and other sources which have two-dimensional gene-gene interaction data. So it is available for rice, but if you look into Arabidopsis or other species, it is also available for some of the other species. And if you click on that node, then what you can see this wheel card type of the structure that is uh, putting the curated protein in the center, and then these are other projected gene-gene interactors. So this way we understand that curating pathways manually is a very slow process, and so gene orthology automated projection help us to scale up to other species, and bringing the data as an overlay like this can also bring the other curated data that is two-dimensional, not as much detail curated as we wish, but our users can have this facility, and if they like to check into some of the other interactors or have insight, it can be provided. Users can also upload their data, whether it is metabolome data, proteome, or transcriptome data, and then see upregulation or downregulation of the genes in response to any of the stress or across the different developmental studies, or if one is comparing the single cell data and so on. So typically plants have polyploidy. We have many gene duplicates, and not all gene copies are expressed at the same way. And so therefore it is a challenge to identify the correct candidate genes, and some of those duplicate genes also have tissue specific expression, developmental stage specific expression. And so in that way, this pathway enrichment analysis allows us to select the candidate genes out of many duplicate genes that might map to the same protein or might encode for the same protein. So this is a little bit about pathway enrichment analysis. I showed you a metabolic pathway, hormone signaling pathway. This is another type of pathways that we have. So this is the how plants recognize bacteria or a pathogen that is from the fungal origin, and what type of the signaling is happening in the plant. So this is a biotic stress response pathway. This is again a transcriptional network that plants can that we have curated, and so it shows the how plants, especially rice, is a submergence tolerant plant and it can germinate underwater. So here is a transcription regulation network of rice during the seed germination and under the submergence, and how it is now connected to the seed germination, also the stress response, and that kind of the curation. So this is other type. And then we have transcriptional networks. For example, this is a flowering time or inflorescence developmental pathway. So we have short day flowering, we have long day flowering, and there are different components as we understand quantitative trait loci, how they contribute to this both short day and long day flowering. And we provide detailed description if there are differences in different species, for example, rice and Arabidopsis. We also provide the references and also what is known and what is different and similar in such scenarios. So overall, what we are doing in recent years, of course, we had chunk of metabolic pathways, but we are trying to connect metabolic pathways to hormone signaling, plant development, tissue-specific expression, and then connecting to the mutant phenotypes, traits, QTLs, and at the same time looking into how the metabolic pathways, signaling pathways, regulatory pathways connect to plant development, or how plants are responding to abiotic or biotic stress response. So, what are the barriers in curating? And so, I like to say that the gene annotation primarily is one of the barriers for curating pathway knowledge. And just to emphasize that, I like to take you to Arabidopsis, which is a model plant and which has been highly curated. So after 25 years of curation, still, as you can see, that 39% of the Arabidopsis genes lack discrete molecular function and they are categorized as an unknown category. They do have some information about cellular component, which means where they are likely to be targeted, or broad biological process, but still the detailed molecular function and their association to a discrete biological process or plant development or stress response, none of that is available as of now. So you can think about other plants that are less studied and which have more complex genomes, more than 50% or even more of the genes are in this unknown category. So one of the challenge for the biocurators is to improve this gene annotation. So how do we do that? So of course, we read a lot of rice literature and Arabidopsis people read a lot of Arabidopsis literature, and there has been great effort to improve annotation. More recently, we have looked into papers described whenever they come into other species to see if those genes or orthologous genes are not described in the model species. So this is just an example of a barley paper, and it has described a gravitropic mutant EGT2. So if you knock out the EGT2, the roots become highly like they lack the spreading and they become very vertical here, hypergravitrophic phenotype they have. And then we looked into the EGT2. It has a single gene ortholog in Arabidopsis and it has a single gene ortholog in the rice. And they are named, in rice, it is a gene of unknown function, and in Arabidopsis, it is known as ATSAM5. And after this paper came up, then somebody more recently, then they just knocked out this Arabidopsis mutant and they confirmed that it has similar phenotype and likely similar function as barley gene. So we borrowed this information and then we named the rice gene also OSEGT2, and we say, okay, it is a transcriptional factor and it is associated with root growth angle formation and response to the gravitropism biological process. So that is one way where we could improve rice gene annotation. Another paper in barley also came to AGT1 mutant, and that mutant has similar phenotype at AGT2, so it has also hypogravitropic root phenotype, and it had also a single ortholog in Arabidopsis, which is kind of named based on the domain, TLP domain, and it has no function, no association with biological process, and same thing is also for rice. And so this study actually helped us to improve the annotation of both Arabidopsis and the rice gene. So this is extending to the other literature, one process. And so finally, we were able to place both AGT1 and EGT2 in a biological pathway that is associated with gravitropic signaling and root formation. We are using other type of transcription. In addition to the literature and published data, we are also now looking into analysis of transcriptome data, looking into co-expression analysis and promoter analysis, looking into other type of the ChIP-seq data, TSS mapping, and then using this information as a evidence to extend curation of some of the pathways. So the other part is that I told that connecting a metabolic pathway to signaling pathways and plant development. So here is an example of stigolactone biosynthesis pathway. Stigolactone plays an important role in gravitropic signaling and suppression of the auxiliary bird dormancy, auxiliary bird, and tillering in rice, which means branching at the base. So number of the branches. So here is the biosynthesis pathway. If you click, you can see the biosynthetic pathway, and then one can also see what is happening in terms of the auxiliary bird suppression. So this is the example how we are connecting metabolic pathways to plant development and signaling. Here I just like to also see like how we can apply it for this knowledge. And I'm taking an example of biotic stress response and disease diagnosis part, but it could also be true for looking into environmental adaptability, looking into which metabolites uh, could impart more abiotic resistance to heat or drought and so on. And some people have used plant reactome data for that and cited us. So there is plenty of evidence for that as well. So, for example, we asked the question if we can use this knowledge graph for developing any diagnosis for the disease, and if it can be used for developing a remote and proximal sensing technology. So we collaborated with Seth Murray and Art Verhoff at Texas A&M, and we received a small grant from DARPA. And what basically we used, we have the knowledge graphs that we have curated from rice and projected on maize. And then we tried to combine the data that came from proteome and metabolome and transcriptome and looked for the signature and potential biomarkers that can be used for disease diagnosis, and then develop a technology that can sense those biomarkers if it can be applied in the greenhouse or the landscape, monitoring of the landscape using some of the UAV imaging and remote sensing methods. So typically what we have knowledge graphs, and then we had uh, some of the uh, data that highlighted that in when plants are attacked by pathogens, typically the data that we used was bacterial pathogens. Then we identify a couple of pathways that are upregulated. And And then we looked into uh, some of the predicted metabolites, and we see if we can detect the increase in those metabolites or decrease in those metabolites using Raman spectroscopy. And we were able to do that, and then we validated these results in the greenhouse condition. So there is a paper that we have uh, uh, described this concept, and this work is ongoing. So similarly, as I showed you, we had this pathway, biotic signaling pathway, one of the pathway, and it describes in details how plants are sensing uh, pathogen attacks. For fungi, they actually sense the chitin, that is the outermost part of the fungal cell wall, and that kind of induces the response, defense response in plants, and its immunity is activated. So there was another study that was came from Nam Haichua's group and was published recently. And so what they did, actually, they took the Arabidopsis mutants that were defective in these receptors that are sensing the chitin. And then they infected those plants, and then they looked into Raman spectroscopy and saw if they could have some peaks that are available in wild type but not in the mutants. And so kind of they also showed that Raman spectroscopy can be used for detecting the fungal disease. So we showed for bacterial, and later this paper also came up. So it shows a great opportunity to combine knowledge graphs with other type of the emerging technology, and have some of the innovations in learning about the landscape changes, disease threats, and so on. So I like to acknowledge a team of undergraduate students who have worked with me since 2012, 2013, and they have been responsible for reading a lot of papers, entering data, curating pathways, writing some of the scripts. So, without them, I can't really do this work. So, I like to acknowledge each of them. And overall, Plant Reactome, uh, in recent years, we have uh, increased the species in past 20 years from 82 to 139. We have more curated pathways, revised pathways, linking pathways, and basically we are representing 56 plant families. We have about 8,000 unique users per year, and we have Plant Reactome papers put together in the Google Scholar as well. In future, we like to expand pathway curation for major crops and not only using rice as a reference crop, also use AI for enhanced biocuration and integrate single cell in spatial omics data, and then develop tools for genotype to phenotype production. I like to also thank Plant Reactome team at Oregon State, Pankaj Jaiswal and we co-lead Plant Reactome, and then we have Justin Elser as a software developer and Parul Gupta, another biocurator. And we also acknowledge funding from USDA ARS. We received also past funding from NASA, DARPA, and NSF. We also acknowledge in-kind support and contributions from the Human Reactome Project. Typically, we are very, Peter Istachio is one of the curator from the Human Reactome who is our mentor. And then we also acknowledge some of the undergraduate research funds that were provided by OSU. And we have other partners, as I say, that Peter Istachio from Reactome Project, and then also Lincolnstein and Joel and Guanming Wu from OICR, and the Gramene folks from Cold Spring Harbor, led by Doreen Weir. And I described the work we did with the Seth. So I thank you all, and if you have any questions, then I'll be happy to answer.
If there are any questions, feel free to unmute yourself and talk, or raise your hand, or put your questions in the chat. I can start. So one question I had was, if we have transcriptome data, especially from rice, because it is the most well curated, what goes as input to the database? Is it like the TPM values of the different genes, and it automatically maps the expression onto all the pathway representations?
Yeah. So what we have, like any data that is with the EBI, so they have currently 25 species data in EBI, and so that mappings and correlation we have built, and so anytime there is a data available for tissue specific expression will show up in the pathways.
Can users upload their own data and
Oh yeah,
visualize
yeah.
it?
So users can upload their own data. They can go to, data analysis, and they can upload their data and conduct. And we don't see that data. So only users will see that data. And they can save that, and we don't see that. So yeah.
Any other questions?
Hey, Sushma. I was just wondering, can we use this to look at lineage specific metabolic pathways and compare across multiple species?
Oh yeah, yeah, yeah. We can, yeah. So species comparison tool, I did not show you here, but actually I can share, just a minute. I can share the live and show you how to do that.
Sweet, thank you.
Yeah.
I can explore too a little bit more.
Yeah, sure. And so there are two ways to do that. So right now, can I share my screen? Let me see.
Yeah, you should be able to.
Yeah, if I can share my screen. Yeah. So the way one can do it, just a minute. So you go here like analyze data, and then here one can like analyze your data, but you can do also species comparison,
Oh,
right?
I see.
And then in the species comparison, you can do like one by one data. So for example, if you want to see like, uh, say maize comparison, for example, so let's say GM maize, and you say go. And then again, it says how rich is the kind of a projection, and you can increase or focus on certain area. And so we have quite a good projection for maize from here. And then on left hand side, you have broader category of the pathways, but you can, kind of open those, and they're like Chinese dolls, how you open these pathways, right? And so metabolism and things like
Mhm.
that. So, and you can close them. So this is how we browse it here. And then you can choose like a carbohydrate metabolism, and then it is kind of again broader category, and then you can select maybe a particular, say, sucrose biosynthesis, and typically these are the views that you will get, right? So this is the sucrose biosynthesis pathway in rice and maize conserved. But what you see here is that, for example, this particular enzyme glucomutase,
Mhm.
you see half yellow and half blue. And so, which means that the number of the genes that are encoding this particular enzyme in maize and rice are different. And so there is like one to many, or two to many relationship that you have. And then you can click further, and then you can see, okay, two genes in maize are conserved, in rice too, and there is one maybe extra gene. So either way, you can see that also the mapping of the discrete genes and how it is. So you can do that for any other species also, like say beta vulgaris, and then again one can see the conserved genes and things like that. So for doing the phylogenetic comparison,
Yes.
one can bulk load all the data that we have and create that. And actually, I'm working on that manuscript, and can share with you if you like to contribute or have some ideas. So then you can bulk download, and I can actually show you where is that in another presentation. So we had like all phylogenetic this thing. So we have Synacocystis, which is single prokaryotic organism that can do photosynthesis, and it has only 3,000 genes, right? And so some of the metabolic pathways are all conserved in plants, but many are missing, and then there are lineage-specific diversification.
Pretty cool. Thank
Yeah,
you. I was also more thinking of evolutionarily conserved,
yeah, yeah, yeah.
comparisons,
Yeah,
identifying.
and not only the pathways, but also the branches, right? The reactions, how same product can be made through many reactions and parallel reactions, and what is their distribution in terms of the developmental pathways and things like that. So I think together, putting a lot of transcriptomic data and this knowledge graph, we can figure out that.
Thank you for an elaborate answer.
Yeah.
That was helpful.
Lars, you have some
Yeah,
question?
yeah. I was wondering if you could also interact with the database using metabolomics data, and what kind of structure that data would need to have. Would I already have to have, like, for example, annotations for certain metabolites, or can I even use mass features to map them somehow to the database?
So you can have a simple tab delimited file. Your data can be organized, and so your X column, the first zero column can have, you know, uh, the IDs of the metabolites. So we can use PubChem IDs, and we can use KEBI IDs.
Okay.
And then you can have like your samples, right? So samples can be your leaf, root, or something, or it could be treatment, and they can have the quantitative, either quantitation of those metabolites, which have the PubMed ID, and you can upload. So typically for the proteins also, you can have UniProt ID, and then you can have that proteomic data. If you are using the gene expression, then you can have the transcriptome ID or gene ID.
Okay.
Yeah, yeah.
Okay. Thank you.
Hi,
Hey,
any
Dr. Suma.
questions? Yeah.
Yeah, one more. Really nice to meet you, and thank you very much for your talk. It was very interesting. Uh, well, I'm right now a lab tech in Dr. Gora's lab, and I am working also doing some kind of knowledge graph relations from metabolites. And I just wanted to ask you, like given the very big size of all Plant Reactome, how difficult was to kind of normalize the predicates for relationships between genes and other kind of entities. Uh, yeah, how have you managed to normalize those without like diluting the meaning of the predicates and relations?
So I think we use a couple of different things. So first of all, we use the most current gene annotation that is available with Plant Ensemble and Gramene. So we are also part of the Gramene database. So the upper level annotation or the gene ontology annotation or gene model corrections as it happens, we consume every time it is done with the Ensemble and Gramene. So that part we don't do ourselves. What we do, we go further down to Go annotation, and we work on more like gene naming, gene discrete molecular function assignment. And anytime we do that, we share that data back to Gramene, back to UniProt, so that that information gets corrected also in other public databases and links to our data. So we kind of take something and we also give something, uh, and then the data that we curate, uh, how one gene is or protein is interacting with other protein, or if transcription of a particular gene is upregulated by a transcription factor, or if two proteins are dimerizing and having an enzymatic activity, or phosphorylation, or any kind of reaction, then we provide evidence code. And so our evidence code can be like, which has uh, evidence code like experimentally verified using which technique, right? So fluorescence assay, or say, for example, two hybrid, or co-localization, or co-precipitation, or something like that. So we have experimental code and explanation how this information was derived. Sometimes we have also computational inference, and then we say, okay, this information is based on computational inference. Sometimes in the papers, we say that, the orthologous gene, for example, from barley is well characterized, and there is only one or two orthologs in rice, and this is a high probability. So it is a curator's comment. And so we discriminate on those three inferences, how we are assigning every reaction or every interaction in plant reactor.
Okay, thank you very much.
Any other last minute questions? If not, let's thank Sushma for this wonderful talk and for giving such detailed answers. And thank you again for accepting this at the last minute. And yeah, it was great to know about these different databases that exist.